Good evening, everyone. Good evening, everyone online. So yeah, thank you for coming to my lecture today, where I will be talking a little bit about the funky skeletons of sharks and their alternative to bone, that of cartilage. So I thought I'd start with a little bit of kind of who I am, to kind of introduce myself. So my name is Hannah Byrne. I'm originally from County Down in Northern Ireland. And I've always had a long-term fascination of the sea. So as a little kid, I was in rummaging in rock pools, and I was really fascinated in how everything kind of interacted with one another. So how the physical and biological elements came together. And after a visit to Bangor University when I was 16, I was very set that I was going to go there to study marine biology and oceanography. And I did. So I did an MSci degree. So that's a 4-year integrated master's, and had a great old time. And it was during my 4th-year project, my master's project, that I kind of got on the slippery slope towards paleontology. So this was part of a kind of international collaboration of different researchers looking into drivers of the fish tetrapod transition. So this is when the first terrestrial vertebrates came onto land. And specifically, we were looking at whether tides could have been playing a role in this. And so my contribution to the project was to run numerical simulations to see if large tides did occur during the time period where the fish tetrapod transition was occurring. And I became really fascinated in the fossil record of the fish tetrapod transition. I have to say my perception of paleontology at that point was quite ignorant. All I kind of thought it was dinosaurs. And one of the collaborators, a researcher based in Sweden, he happened to have material that needed described and looked at. And so there I embarked on a PhD in evolutionary organismal biology at Uppsala University in Sweden. And there I was very fortunate to study some tetrapod body fossils, but the predominant part of my project was fossilized poo, so coprolites. So that's this kind of bottom image here. So we were using high resolution scanning techniques, and then using it in 3D imaging software to reconstruct the contents, and then later down the line kind of do some ecosystem dynamics studies. So that was a lot of fun. And then some of my colleagues were embarking on a study on the evolution of cartilage. So evolution of cartilage in a whole host of different animals. But they were primarily focusing in on the uncalcified cartilage, so cartilage that we have in our nose and ears. And I remember one of my colleagues saying, oh, but it would be really cool if someone could look at the calcified cartilage in sharks. And I was like, oh, please tell me more. So that's kind of accumulated into my current project then, looking at the evolution of calcified cartilage in chondrichthyans. So this is the kind of group term that we have for sharks. And I'm based at Naturalis Biodiversity Center in the Netherlands. So if we look at the makeup of the chondrichthyans, so the chondrichthyans are comprised of 2 main groups. So the first group we have are the elasmobranchs. So these are the ones that we kind of think about or come to mind when we talk about sharks, skates, and rays. And so you have a whole host of different morphological variability. They come in a lot of different sizes, right down from about 20 centimeter into up to 18 meters. And they can be found in all our oceans and at kind of various parts of the water column. The kind of lesser well-known group are the holocephalans. The holocephalans, as we can see here, are comprised of the elephant shark and the ratfish. So these are kind of smaller, deep-dwelling organisms. So as I mentioned, we find our chondrichthyans right across the water column, and the deepest kind of record of a shark is the Portuguese dogfish. So we're getting right down to 3,700 meters. And I thought I'd kind of do some fun facts on some cool chondrichthyans. So the oldest living vertebrate that we know of, the Greenland shark, is postulated to live up to 400 years. It happens to be my favorite of the sharks. And another couple of fun facts about Greenland sharks. So 98% of them are blind. So there's a parasitic copepod that attaches itself onto the eye. And they're only able to differentiate between light and dark. So imagine going around for 400 years, only be able to see light and dark. And there also was a case where one was choking on some moose skin in Canada, and a couple of people had to drag it out of its mouth. So there you go. So that's the funky Greenland sharks. The oldest record of a fossil fish, so a fossil jawfish, is a shark, a chondrichthyon. So we have some scales here from Australia, and these are dated to 455 million years ago. So really, the chondrichthyons go, really far back in our history. And this is really interesting because trees didn't make it onto land until about 400 million years ago. So we have, sharks kind of cruising around our oceans about 50 million years before trees. So for a whole host of reasons, they're a very charismatic group. They play a very important role in our marine ecosystems, and they've survived all 5 major mass extinction events in Earth's history. And I believe one of the kind of key reasons why they have been such a successful group lies in the composition of their internal skeleton. So if we look at a very basic phylogeny, so yes, I'm not talking about jawless fishes in this talk, I'm talking about the jawed fishes. And these can be split into 2 groups. So the chondrichthyons I've mentioned, but we also have the osteichthyons that include ourselves. And the major kind of differentiation between these 2 groups is the makeup of the internal skeleton. So osteichthyons have an internal skeleton made primarily of bone, and the chondrichthyons have one made completely of cartilage. And for a long time, having a cartilaginous internal skeleton was believed to be primitive. This is based on the idea that evolution would be driven towards more complex tissues like bone. And also the fact that kind of early in our life stages, a lot of our skeletal elements start off as a scaffolding of cartilage, and then this is replaced by bone. However, if we delve into the fossil record and we go to a group of extinct fishes that make up the kind of last common ancestors, both osteichthyons and chondrichthyons, we have the placoderms. And researchers, including Naturalis in the room, they discovered the presence of endochondral bone in a skull roof element of one of these fishes. So this is the endochondral bone is what we have. And so this is really interesting. So this is showing the presence of endochondral bone before we have the split of the chondrichthyons. So not only is their internal skeleton not primitive, they decided they wanted to move away from bone, and they evolved a complete suite of cartilaginous tissues to replace the replace the properties of bone. And this kind of makes them the aliens among the vertebrates. So what are these suite of cartilaginous tissues? So if we look at this beautiful scan here, if we kind of focus in on one of the skeletal elements here from the jaw in the circles and in B, we can see that the skeletal element is covered by lots and lots of tiny discs, and these discs are calcified cartilage, and each of these discs are known as tesserae. And collectively, the tesserae make up the tessellated calcified cartilage. So from now on, I will be talking about the tessellated calcified cartilage as the TCC. And each of these discs of calcified cartilage are typically pelagonal in shape. They're interconnected to one another by spokes of calcification. And the little gaps in between are the intertestral spaces. And if we look at D and if we look at images D over here, we can see that this is actually a very thin layer. of calcification. And internally then we have an uncalcified cartilage core. And this uncalcified cartilage core is very similar to the cartilage that we have in our nose and our ears. And this layer of tessellate calcified cartilage is overlain by a fibrous layer known as the perichondrium. We also have another unique calcified cartilage in chondrichthyans. This is known as the areolar calcified cartilage. And you find this exclusively in the centra, so in area C here. So I will not be talking about this anymore. The kind of focus of my studies in the talk will be on the tesserae, so the tessellated calcified cartilage. And if we go in a little deeper into these little tiles of calcification, so we're looking at the ultrastructure. So here we have a virtual thin section from the lower jaw of a Mediterranean starry ray. So yes, this is a cross section between two of these discs of calcification. And what we can see are a lot of little blobs. And each of these blobs are known as chondrocyte lacunae. And if we kind of study this in detail, we can see actually there's a differentiation between the shape of some of these lacunae on the top and the ones at the bottom. And this represents two different layerings of calcification. So on top, we have these kind of like elongated cell spaces, and this is the prismatic calcified cartilage. And this is a cartilage unique to the chondrichthyans, so the sharks and relatives. And underlying this, we have the globular calcification layer. And this is indicated by the kind of more globular shaped cells. And here we can see the spokes kind of interconnected between them, and then the spaces there are. So clearly this is a very important tissue for the chondrichthyans. It provides somewhat of an optimum between stiffness, flexibility, but is also super lightweight. So what do we actually know about the evolution of this tissue in fossil forms, but also what do we know in modern forms? So we go back to our phylogeny. So right down at the kind of base of the chondrichthyan stem group, we come across a group of extinct fishes known as acanthodians, also referred to as the spiny sharks. And this is a very interesting group because they have a mixture of both bony tissues, but also cartilaginous ones. So there has been great work done by several researchers who have done 2D thin sectioning of skeletal material from these animals. And they have noted, yes, the presence of bone, but also within the globular calcified cartilage, you start to have these separations. And these separations seem to kind of indicate that they are the precursors to tesserae, so this tessellate calcified cartilage. And they have kind of termed it the subtessellate calcified cartilage. Excuse me, if we move slightly further up the tree, so still within the stem group, we come to a group of fish, including Puccopampella, Doliolus, and Gobacus. And here we start to see the emergence of the tesserae. So we start to have these separate discs of calcification. And this is the first time we see the prismatic calcified cartilage. And it's termed prismatic as when you look at it under black light, you can see the nice kind of prism colors. But we're not quite there at the crown group because we still have some bony tissues in these animals. So now we get into the chondrichthyan crown group, and one of the kind of main defining features of the crown group is the presence of tesserae, so this tessellated calcified cartilage. And we have our kind of two major groups, so the elasmobranchs and holocephalans. So within the fossils of the holocephalans, we can see that the tesserae are nice and distinct. They're pelagonal in shape. And this is generally noted among several fossil holocephalans. If we then look at the elasmobranchs, we can see the same story. So here, this bottom right image, we can see very nicely the spokes interconnecting the tesserae to one another, and also the spacing. But we're also noting some kind of extra features. So here on the top right image, we can see that there are layering of the tesserae. So we have multiple rows of tesserae. And this has also been noted in some modern forms, and this is believed to be present in areas where there is extra stress. So this is kind of giving kind of more of a robust structure in that region. So up until now, we kind of have some ideas of what's going on, but it's exclusively based on 2D data. So we're not getting a 3D perception of these of this tissue, and also we're getting kind of sporadic information from different skeletal elements, so it's hard to kind of make a accurate comparison between them. If we go into the extant forms, into the modern forms, now researchers have started to utilize 3D imaging software. So now we're getting an understanding of what these tesserae look like in 3D. And we look first at the holocephalins here. We can see they have changed in their appearance from what we're seeing in the fossil forms. So now the tesserae seem to be kind of more square in shape, maybe more amorphous. They're much closer packed together, so we can't see these individual spokes interconnecting them. Whereas we look at the elasmobranchs, they have retained this kind of polygonal shape on the spokes. So here we have 4 images. So the two on the top here are from a couple of skates. Here we have the bottom left is from a hoard shark, and then the bottom right here is from a deep dwelling 6-gill shark. And in general, yes, we can see the polygonal shape and the interconnecting spokes. So we're starting to gain an understanding of the kind of variation between different modern chondrichthyans, but this technique has only been applied to just over a handful of different taxa. So we're still missing the kind of variation across all the sharks, skates, and rays. So that kind of brings me to kind of the purpose of my project, and that's to make a concerted effort to kind of apply this 3D visualization technique, not just to kind of more groups or more fish from modern forms, but also to apply that right down into the fossils. And the aim to my project is to look at the 3D histology of the cartilage, both in modern and extinct chondrichthyans. And the skeletal element I focus on is the lower jaw. There's a couple of reasons for this. One of the primary ones is that it tends to be kind of readily available and identifiable in the fossil record. And it's also something that I can look also into whether variation in the tesserae and its arrangement is maybe related to different feeding modes. And I thought this was a kind of a good point to discuss why is this work important, other than sharks are cool, they have funky skeletons, we should understand it. So within the realms of paleontology, it's super important for our understanding of evolution of hard tissues as a whole. A lot of focus has been on bony tissues. And so this project will kind of play a role in kind of getting us up to speed on the evolution of cartilage, but also kind of understanding how the cartilage in chondrichthyans and sharks, how that relates to cartilage in the likes of us. And also kind of in this transition away from bone into cartilage, then how bone relates to cartilage and how these different mineralization processes are similar / different. In terms of marine biology, as I mentioned before, sharks play a super critical role in our marine ecosystems. And so it's important that we get a better understanding on the kind of architecture of their skeletons and also these kind of functional adaptations that they have with different feeding modes. There are applications for engineering. So some of you may have seen that I think Lufthansa have utilized inspiration from the denticles of sharks to reduce drag in their airplanes. And indeed, this kind of biomimicry can also be possible with these tessellated structures. So as I mentioned before, they're quite lightweight and also give this degree of both flexibility and stiffness. And then there are also the kind of medical applications. So one of the big struggles that we have with cartilage cartilage diseases such as osteoarthritis is that the cartilage kind of spontaneously calcifies, and we don't really understand why, nor do we understand the mechanisms behind this calcification. But obviously in chondrichthyans it's a very purposeful and very important part of building their skeleton. And so I am collaborating with researchers at Leiden University Medical Center to kind of gain a better understanding of how the cartilage and how the calcification mechanisms that we see there, how that differs or may be similar to what we see in the sharks. So my project can kind of be broken into three general parts. So usually in paleontology, we start right down with the kind of earliest fossils and we work our way up. However, I've kind of flipped it on my head. So I'm looking at the extant modern forms first and then going then back into the fossil record. The reason for this is that as I've demonstrated, that we don't even have a good understanding of the variation of tesserae, both in their shape and arrangement in modern forms. So it's good to get a kind of a good solid foundation from the modern forms before then tackling the more challenging fossil forms. And this will be utilizing high imaging techniques, which I'll talk a little bit more in a second. So we're looking at the kind of overall architecture of the tesserae in both the upper and lower jaw, but then also delving right into the kind of histological details, so at the cellular level as well. And then I will apply the same techniques then to some of the fossils. So here we have an example of one that I've scanned from Edinburgh. And here's a sneak peek at some of the beautiful fossilized tesserae. And then hopefully with kind of all of this knowledge, then we can start looking at the kind of more challenging material in the stem group, so in the spiny sharks, acanthodians, where we have a mixture of these bony and cartilaginous tissues. And this is kind of a very rough pipeline of what I do. So I have specimen, put specimen in scanner. Scanner gives me a stack of images. And then these images are imported into 3D imaging software where I apply masks to the areas that I want to visualize and segment out. And then up pops a really nice 3D model that then I can study and interpret. So the material that I've been working with, I have been very fortunate to have loaned material from these different institutions. So in my data set, I have a nice even split of both modern taxa and fossil taxa. So we have 16 modern taxa, and these are from Naturalistic Museum in London and also at Naturalis, where I'm based. The majority of these were micro CT scanned, but also synchrotron scanned, which I'll talk a little bit later on. And then I also have 16 fossil taxa, which primarily come from the NMS in Scotland, the Museum of Natural History in Paris, and then also New Brunswick in Canada. So the first scanning technique I want to talk a little about is micro CT scanning. So some of you in this room who have unfortunately maybe broken something in their body have likely gotten a micro CT scan. So I'm very fortunate to have the use of a micro CT scanner at Naturalis in the Netherlands. And so we put many of our extant specimens in the scanner. So here we have, a very wrapped up and sad looking cookie cutter shark that was put in there. yeah, and from this then I'm able to visualize the tesserae in 3D. But if we want more kind of high resolution, so the image that I showed previously of the kind of cross section through the two tesserae, then we need to go to somewhere that has a little bit more oomph, a little bit more power. And this brings us to the European Synchrotron Radiation Facility. So this is essentially CT scanning, but on steroids. So here, nestled at the foot of the Alps in Grenoble. So this here is an electron ring. So it's essentially a bit of a mini CERN. So this ring here is almost 1 kilometer in diameter. And there is a nuclear facility there that powers this ring. And what's going around in the ring are electrons. So electrons are flying around at very high speed. And then coming off at tangents are what are called beam lines. And at these beam lines, then this is where the scanning takes place. So the electron ring is disturbed by magnets, and this produces photons. And the photons are what we use for the scanning. So for scanning my project, we used 2 beam lines, BM5 here and BM18. Now, the specific scanning technique we use is a bit of a mouthful. So it's propagation phase contrast X-ray synchrotron microtomography. And what's quite unique about this method is, so instead of just measuring the absorption of the rays it passed through the specimen, the kind of the phase shift of these of the rays are recorded. And what that enables us to do is to really get very fine resolution of the internal features of our specimen. And also if we have specimens where the density differences are maybe poor, so if we're looking at soft tissues, or in my case, looking at the cartilage, which tends to be not as heavily mineralized, then we get a much better detail. So here on the bottom right is an image showing here on the left, just using the kind of conventional absorption, and then on the right showing then the use of the phase contrast, so getting much sharper, much detailed features. So the specimens were scanned over 2 different sessions. So as you can imagine, the ASRF is quite a big facility. It's also quite an expensive facility. So if you were to pay yourself, you're talking maybe €20,000 for scanning in a day. But fortunately, they do have funded rounds. So we were fortunate to get 2 scanning sessions funded. So the first session was led by Dr. Sophie Sanchez and colleagues, and they scanned a number of specimens, both BM5 and BM18, and they did spot scans at different resolutions. And it's here this kind of what we call sub-micron, so not 0.8 micron is then when we start to get the cellular details of the cartilage. And for this study, they did a single spot scan in the lower jaw. And the second session, so we went back in April 2025, a session led by myself. And we scanned, we basically increased the data set. And we also increased the areas in which we scanned. And this was to capture the kind of variation of the tesserae along the lower jaw. Because from my work with the CT scans, I noticed that it's not homogeneous. So the tesserae don't look the same at different points of the jaw. And this was an interesting learning experience. So good to take small specimens and specimens that don't have much rock around them. And this is just to show the comparison of a CT scan versus a synchrotron scan of a fossil that I scanned. So this is one of the kind of earliest sharks. So this is Doliotis from Canada. And here on the left, we have an image from a CT scan taken from the US. So we can see that, we see some teeth and we see some other mineralized tissues. So in terms of you're doing kind of morphological descriptions, this scan is perfectly fine. However, we want to delve into the kind of cellular details. And so this is where using the synchrotron, this phase contrast element, gives you much sharper detail and more, yeah, you get more detailed information out of it. Ah, yes. So this is a little kind of fun part of the scanning at the SRF. So when you get your allocated scan time, you're going the whole time. So if it's 4 days, you're working day and night. So you tend to take it in shifts. And we acquired a little mascot for our scanning session. So our mascot here, the little teddy bear, was named Dolioli. And it was named Dolioli after a colleague kind of mispronounced Doliotis. And yeah, so Dolioli had a great time at the SRF. And we made, yeah, little cardboard cutouts for it. And Dolioli even went on holidays after. So Dolioli had a great time in Ireland after the scanning. So we'll start getting into some of the kind of exciting outputs from the project. So my projects kind of be classified into 4 different studies. So again, looking at the variation of the TCC in modern forms, then looking at the ultrastructures, the cellular details, then doing the same for the fossil chondrichthyans, so the fossil sharks, and then finally delving into the precursors in the STEM group members. So I'll show you data from study 1. So this is a study that I'm writing up at the moment. And then I'll show you some work in progress from study 3. So yeah, so study 1, a lot of the work was conducted by a great undergraduate student who did an internship with me last year, where we looked at a bunch of modern sharks from the collections at Naturalis. So the kind of primary purpose of this study was to get a good kind of phylogenetic spread of different modern sharks. So here we have the kind of 4 major groups represented. So we have the batoids that have the skates and rays, we have the galeomorphs, we have the squalomorphs, and we have the holocephalans here. And one of the kind of caveats of this study is that we worked with smaller specimens, and this was due to the restrictions of the micro CT scanner. But this gave us a good opportunity to look at the kind of early development of the test ray, the TCC, and indeed it was quite well developed in most of the forms, so we could see it in the CT scans and make nice 3D models from them. What we also wanted to do was to look at a variety of different feeding modes, and also sharks kind of feeding on different diets. So here's this kind of information summarized. So in terms of the feeding modes, so the 3 primary kind of feeding strategies that we have in the chondrichthyans are bite, which we're quite familiar with jaws and the likes. And we also have suction feeding, and suction feeding was the kind of primary mode with a lot of the sharks from our study. And you also have ram feeding. Now, ram feeding tends to be associated with the filter feeders, so the whale shark and the basking shark. So given their size and actually the lack of specimens, they weren't included in our data set. And yeah, we wanted to also look at the role of diet. So one of the kind of main influences on the variation of the TCC that a couple of studies have looked at is the role of sharks feeding on hard-shelled organisms. So this is called jurophagy. And so they've noted that there is specialized features in the jaws of sharks who feed on hard-shelled organisms, which I'll show shortly. So now I'm going to show you what we have nicely here. So we took the scans of these sharks and Isabel segmented out the upper and lower jaw on one side. And these are the results that I will show you. I'll not show you all the specimens. I'll show you kind of a little, the most important ones. So we have the upper jaw represented in blue, and we have the lower jaw in pink. So here we have Xanabatus. And kind of right off the bat, we can see these beautiful polygonal tesserae. We can see very clearly the spokes connecting them together, and we can see the spacing. And if we look at the upper and lower jaw as a whole, the appearance of the tesserae is quite the same, so quite homogeneous across the upper and lower jaw. However, there are some areas where you have weaker mineralization. So these are typically in areas of articulation, so where they're interacting with other skeletal elements, as we have here. What you'll also be able to note, it's maybe not so clear in some areas, but in these zoom ins we can see these holes. Okay, and these holes are different from the intertestral spacing. So these holes are pores. Now, the role of the pores is kind of poorly understood in the chondrichthyans, but what most of them tend to lead to are these invaginations of calcification. So if we can look here, so we're looking internally in the lower jaw, and we can see this kind of bridge of calcification. So it was going from kind of one layer of tesserae right across to the other. And this is known as trabeculae. And these trabeculae have been associated in areas that are dealing with kind of high strain. So in these sharks that are feeding on hard-shelled organisms, you tend to find these trabeculae. And this is the story for pretty much all the batoids, so all the skates and rays that we looked at. The tesserae are well developed, polygonal in shape, and we have these nice interconnecting spokes. Now, if we look at Ginglimistoma here, so this is a little nurse shark, it's quite a different story. So the appearance of the TCC, of the tesserae, it varies quite a lot depending on where you're looking at on both the upper and lower jaw. So we have some regions where we're getting polygonal tesserae with interconnecting spokes. However, the tesserae are quite small. And you have quite a variation in the level of mineralization. So you have some regions kind of here and here in the upper jaw, which almost have no mineralization at all. And also in the kind of internal surfaces. Excuse me. But what you can see quite clearly and are quite numerous are the pores. And what is really fascinating about this specimen that we can see quite nicely in this scan is that there are numerous invaginations of calcification. And in many cases, these are going right across the other side. So I visualized it here for you. So we've kind of cut the upper and lower jaw in half. And we can see numerous bridges of calcification. So predominantly they tend to be in the kind of more anterior, so the front parts of both the upper and lower jaw. But if we look here at the back of the lower jaw, we can see this kind of humongous kind of tree branching almost of the trabeculae. So this is really interesting to see in such a young nurse shark, to see such extensive calcification and trabeculae. If we look at another member of the galeomorphs, we see a very different story. So the mineralization is actually very poor in this specimen. So this is a smooth hound, and we can see that in the scan. So the contrast of the calcification is quite weak. And it appears almost as like a very thin veil of calcification where we can't see individual tesserae. And in quite a few areas, we have very large areas where it's very poorly mineralized. So at this young life stage, the tesserae are very poorly mineralized. Now we go over to the squalomorphs. So we have an angel shark here. And this is more similar to kind of what we're seeing in the nurse shark, maybe actually kind of more heavily mineralized. So as we can see from the scan here, we have beautiful contrast between the calcified cartilage layer. And in some regions, we have indeed the nice pelagonal tesserae with the interconnecting spokes. But we also have areas where the calcification is so prominent, so the mineralization is so high that the tesserae kind of merge into one another. And we also have numerous pores throughout. And these pores, some of them do lead to these invaginations of calcification, as we can see here in eye, but these do not kind of propagate the whole way across. And we do have this area where it's not as highly mineralized. Now we move over to this fun little lads. This is a cookie cutter shark. So this is the only adult that we have from our data set. And interestingly, the upper jaw of this animal is so poorly mineralized that you can barely see it in the scan. So it couldn't be segmented out. So we only have part of the lower jaw. And what we can see here is the tesserae are quite amorphous, so they don't really have quite a distinct shape. We don't have interconnections between them, which is quite odd. And the level of mineralization varies quite a lot. So kind of towards the more central regions, we have higher levels of mineralization, but here in the kind of ventral on this bottom side, it's very poorly mineralized. And then we go on to our solo holocephalin of the group, this chimera. And this looks very kind of distinctly different from what we've seen from our other sharks. So you don't see kind of individual tesserae per se. It kind of, the calcified cartilage takes on a more kind of lattice-like structure, and it gets very wispy in some regions. And you have this very large area on the internal surface that is, it appears to be unmineralized. And if we look at the scan here on the left, so the holocephalins are quite unique in that they have tooth plates rather than teeth. And this seems to line up with so the area where we have very little to no mineralization lines up with the kind of interaction then with that tooth plate. So how can we explain these observations across these different sharks? So firstly, is there kind of a phylogenetic signal? I think there is for the batoids, so the skates and rays, so there seems to be this quite robust signal that early in its life stage, so early in ontogeny, the tesserae is already quite well mineralized, pelagonal in shape, and this seems to be homogeneous, the same across the jaw surface. And for the other groups, we have quite a lot of variation between the different animals. Is there an ontogenetic signal? So kind of different life stages. We would need to look at more of these organisms at different life stages. But again, with the batoids, we seem to have this kind of early onset of the tesserae being well mineralized. Could it be due to the different kind of feeding modes? So we have kind of suction versus biting. Among our suction feeders, it does seem to be that the tesserae is well mineralized and developed early in ontogeny, whereas if we compare it to that smooth hound that we looked at, which is a specialist bite feeder, it's quite poorly developed early in its life stage. And I have looked at scans of an older individual where the tesserae is more well developed, and actually you get multiple layering of the tesserae. Does diet play a role? Again, and this is something that I would like to look into further, but of course you do have diet shifts depending on the age or life stage of these animals. But again, there does seem to be, with organisms feeding on hard-shelled, of sharks feeding on hard-shelled organisms, they will have these specialized features, like the bridges of calcification going across the internal parts. Body morphology, is that a contributing factor? So the batoids are all quite dorsal ventrally flattened. And interestingly, two other sharks that we looked at that have quite well developed tesserae early in its life stage are the angel shark and the nurse shark, which are also dorsal ventrally flattened. So maybe that's also a reason. And then finally, is there other kind of ecological factors at play? So does depth have a play a role? So two of the sharks that we had are deep water taxa. So we have the cookie cutter shark and the chimera, the holocephalin. And it has been noted that generally these sharks tend to have less well mineralized cartilage. And I would like to kind of investigate this further. However, you do have outliers like the smaller spotted cat shark, which also has poorly developed cartilage, but it tends to be more of a shallow water dweller. And then finally, kind of saltwater versus freshwater. So we had one freshwater shark in our group, Potamotrigon, but so it belongs to batoids, the skates and rays, but it seemed to be quite similar to its saltwater relatives. So in conclusion, the batoids rule. So the batoids have very well mineralized tesserae early in its life stage. But we also see it for the nurse shark, the angel shark, and the bamboo shark. So is it because they're suction feeders, or maybe does the body morphology play a role? There does seem to be a strong signal that the suction feeders have well-developed tesserae early in its life stage compared to bite feeders. And there's something quite extraordinary exhibited in this nurse shark, that even the very closely related bamboo shark does not exhibit such extensive internal mineralization. And the deep water taxa we looked at seem to mineralize their tesserae less, but also so does the cat shark. So need to look into that further. All right, so on to the second study. So this is looking then at what's happening in the fossil forms. So this is some kind of work in progress. So this is mainly based on the scan data. So no 3D models just yet. So if you look at the scans of two stem holocephalins here, so we have Acmenistian and Chondrochelys. So these are both from Scotland and date to, I think, around 340 million years ago. And what I'll show you is a video running through the scan. So we're going from the external surface right through the center and then out to the external surface. And these specimens are quite flattened. So yeah, so you might not see the kind of differentiation of the two layers as well. So if you look at Acmenistian here, so it kind of looks like a really nice kaleidoscope. But what I want you to draw your attention to are these dark structures here. So these are the tesserae. You can see really quite nicely preserved. We have the pelagonal shape and we have the interconnecting spokes. So this kind of concurs with other observations on the fossil holocephalins that show these nice pelagonal tesserae. Whereas we look at Chondrochelys here, we're looking again at these dark structures. And the shape of the tesserae seems to be different. So it seems to be more kind of amorphous in shape, and we don't have those nice interconnecting spokes. So if we look at a summary of this, so yeah, so this is quite interesting because this is quite kind of contrasting appearances of the tesserae quite early on in the holocephalin lineage, with the left here being kind of the more condition we see in modern elasmobranchs, and then on the right, more akin with what we see in modern holocephalins. And then if we look at example of two stem elasmobranchs, so we have Hybotis here on the left and Tristichius on the right. Hybotis is from the Jurassic of England, and Tristichius on the right here is from the Carboniferous of Scotland. And here we're looking at a cross section through the jaw. So we have the two layers of the tesserae. And what I want you to draw your attention to are these extensions of calcification going right through the uncalcified cartilage. And this is very similar to what we've seen in some of the extant forms. So these are the trabeculae, these bridges of calcification, which is really neat. And we can see nicely the individual tesserae, which are kind of stacking on top of one another in some areas. So you are getting some layering of the tesserae. Now, if you look at tricycus here, so a little less clear. So we're going from kind of the front towards the back end. And again, you're seeing this multiple layering, but kind of much more drastic in tricycus. And it seems to be multiple kind of monolayers. So what you can get is a polylayer. So that looks kind of more like how you'd build a stone wall, which is kind of what we see more in Hybotis. But this seems to be kind of multiple layers of tesserae stacked on another. And if we look into the feeding modes of both of these, so Hybotis from the teeth morphology is said to be a durophagous feeder, so feeding on hard-shelled organisms. And these features of the bridges of calcification, these trabeculae, also match up with this. So this is a feature specialized for its feeding. And then with tricycus, it is said to be a suction feeder. So again, high stress is being exerted, and so the need for kind of more strength and robust tesserae, as shown here. And then finally, looking a little bit into the fossil batoids. So this is from a scan of Sceliorhynchus. I'll say it's that. And really nicely, we can see the tesserae, and really kind of typically pelagonal, kind of interconnected with one another, and looks really similar to what we're seeing in the modern batoids. And this is from the Cretaceous of Lebanon. And then finally, we have Bellum novatus here, so this is from late Jurassic of France. And these are the tesserae. And what we can see here are cell spaces. And this is really neat because it has been said that in the skates and rays, there's an increase in cellularity. So there's an increase in cell spaces in the tesserae of batoids compared to the other sharks. This fossil kind of pushes back the origin of this increased cellularity to the late Jurassic. So to kind of wrap up what I've just been talking about. So kind of what do we know now and what's kind of left to do? So we've established that these scanning techniques work really well for visualizing the calcified tissues in chondrichthyans, both in modern and fossil forms. We've increased our understanding of the variation of the tessellated calcified cartilage in modern forms. And we are seeing similar structures or similar adaptations in both our modern and fossil forms. So these bridges of calcification, the trabeculae, we're seeing in modern forms and in fossil forms. And we can link these then to adaptations due to feeding strategies. And then finally, yes, this observation of the increased cell spaces, cellularity that we see in the batoids, we can see in this fossil stem batoid, and this pushes back the origin of this trait to the late Jurassic. So much work has been done, but there's still much more to do. So the kind of the big kind of looming project at the end is what's happening in the stem chondrichthyans. What are these tissues that we're seeing in the spiny sharks, and how is it relating to what we see in the modern forms? I want to increase the data set, so not just for the fossils, but also the extant, because even within extant taxa that are very closely related to one another, have very similar feeding modes, very similar body morphology, the tessellated calcified cartilage differs quite a bit. So it's good to get as much information as we can. And then finally, the development of the tessellated calcified cartilage throughout ontogeny. And not just looking at it in the lower jaw, but also how the development of it is in other skeletal elements. And with that, I have a whole host of thank yous to do. This is something that I couldn't do without all these wonderful people. Yes, and the funding of the Marie Curie. And with that, I'd like to thank you for listening and take any questions.